Hello, I'm Jackie Starrett, Senior Director of In Vivo Pharmacology at Tyra, where I lead preclinical research in skeletal dysplasia. Dr. Will Charlton, our Senior Vice President of Clinical Development, who is leading our skeletal dysplasia clinical group, will join me to provide his perspective on the potential clinical impact of our research findings. FGFR3 is a receptor tyrosine kinase. It consists of an extracellular domain which senses growth ligands, a transmembrane domain, and an intracellular kinase domain. FGF ligands bind to the extracellular domain, causing it to autophosphorylate and trigger downstream signaling, which negatively regulates bone growth. Activating genetic alterations in the FGFR3 gene are responsible for driving multiple skeletal dysplasia conditions in people, including achondroplasia, the most common form of dwarfism, and hypochondroplasia.
Beyond the functional challenges of disproportionate short stature, there are multiple comorbidities associated with achondroplasia, including compression of the brainstem and the upper cervical spinal cord, which can require surgical intervention, highlighting the unmet medical need.
To meet this need, we designed TYRA-300 to target FGFR3 over the other FGFR isoforms, and to inhibit the overactive variants. We began by testing TYRA-300 in wild-type mice, observing dose-dependent increases in body length and in the long bones. This model provided an effective preliminary screen for evaluating compounds prior to testing in transgenic models of skeletal dysplasia. Next, in collaboration with Dr. Leger-Mallet, we tested TYRA-300 in an achondroplasia mouse model carrying the FGFR3 Y367C mutation. Daily treatment significantly increased body length of the mice by 17.9%. We also observed significant increases in long bone length, including the femur and the tibia. The anatomy is evident under X-ray and striking under micro-CT.
While growth is an important indicator of efficacy, we aim to reduce foramen magnum and spinal stenoses, along with surgeries related to these complications.
We observed a significant increase in the area of the foramen magnum, along with improvements in the skull size, both in length and width, making the overall shape more similar to the shape in a wild-type mouse. We next studied the mechanistic effects of TYRA-300 within the growth plate. Looking first at the growth plate in a wild-type mouse, we saw a healthy secondary ossification center, followed by columns of proliferating chondrocytes and then hypertrophic chondrocytes. Histologic analysis of the mouse with achondroplasia revealed a smaller and highly disordered growth plate architecture. After TYRA-300 treatment, the secondary ossification center was restored, proliferating chondrocytes increased and organized into columns, and hypertrophic chondrocytes became larger and more differentiated.
Individuals with hypochondroplasia often have less pronounced skeletal features than those with achondroplasia and are less likely to experience medical complications.
Our studies with Dr. Leger-Mallet showed that TYRA-300 significantly improved bone length in mice with hypochondroplasia. Our preclinical research shows that TYRA-300 increased bone growth across genetic backgrounds, improved cranial structure, and restored normal growth plate architecture.
Currently, the only FDA-approved treatment for achondroplasia is a once-daily injection of vosoritide, a C-natriuretic peptide analog which is marketed as Voxzogo. TransCon CNP is in the process of filing for FDA approval, and infigratinib is currently in a phase 3 clinical trial. TYRA-300's promising preclinical results as a selective FGFR3 inhibitor have led to a phase 2 clinical trial called Beach 301, which is currently enrolling. Our goal is to create a safe oral treatment for children with achondroplasia.